Chapter 6, Section 4, Bidirectional Replication, the Replosome, and Proofreading. Keeping in mind, DNA replication for all living organisms is going to be bidirectional. Okay, you're going to have one origin of replication, and then these replication forks that we talked about in Section 3, that replosome is going to go in opposite directions away from it. You're not just going to have one start and then continuous on from there, because that would take too long. So you have two replication forks traveling away from the origin of replication. For prokaryotes, which we're talking about here, DNA Pol III is laying down about 1,000 nucleotides per second. So in the time it took me to say that, 1,000 nucleotides per second. Bacteria in your body, in your gastrointestinal system, laid down about 2,000 nucleotides. So as you've seen before, I know in Intro Cell and Molecular, and hopefully in genetics, if you've had that already. ORAC, our origin of replication. DNA A, the helicases and everything are going to unwind, and you're going to get the beginning of the replication bubble. You're going to have two replication forks, one there, one right there. And they are going to move away from each other towards this terminus region here. As the new chromosome is being made, it's separated out. So you end up with two complete new double helices. Dark green is to signify original, light green is the new copies. And hopefully you remember, like we talked about last section, leading strand, lagging strand, and then opposite of it across the origin, lagging, leading. It all depends on the orientation. You got to have an open 3' to build on. And if the open 3' is in the wrong direction, pointed away from the replication fork, then it's going to have to be a lagging strand. Now that replosome that was mentioned in the previous sections, this is a complex of proteins. Complex of proteins that their one and only job is to synthesize a new strand of DNA that is an exact complementary copy of the template. No mistakes. You have the primosome, which is also going to be part of this, is the helicase, the primase complex. Helicase unwinding, stabilizing with topoisomerase and whatnot, and the primase laying down whatever primers are needed, leading strand, lagging strand. The opposite end of the origin is going to be the terminus of replication. This is where, when the replication forks run into each other, done. Everything is complete. Once the two new chromosomes are made, FITZ, one of the proteins that's responsible for aiding in the division of the cell, will aid in separating the two new chromosomes to different regions of the cell. So when you look at this replosome, you have DNA helicase unwinding. Here's our Pol III's. We have right here in front, this is a clamp protein that's holding DNA Pol III in place so it doesn't fall off, because if it falls off, replication's done, not going to occur anymore. Primase, right here next to the helicase, is laying down new primers. Boom, 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 boom. Gyrase is up here, stabilizing, doing what it can to get rid of the overcoiling that may happen as everything's unwinding. But notice, in many figures in textbooks, they show leading strand, lagging strand proteins, enzymes, independent of each other. And no, what we found is they all are complex together and held together by a collection of proteins. Here they're showing one of those proteins known as tau. Tau is interacting with the two DNA polymerases, the one on DNA Pol 3 that's on the leading strand, the DNA Pol 3 on the lagging strand. So technically what happens is these are all held in place, and then the double helix is fed through. The leading strand is fed directly through. The lagging strand does this loop out, feeds in, pulls, 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 feeds in some more, pulls it back through, because that's the only way this can work and go through here. So these enzymes and proteins stay in place. It's actually the DNA that is fed through and is moved around this replosome complex of enzymes and proteins. However you look at it, when it comes to proofreading, prokaryotes are pretty much error-proof. Not quite as well as us eukaryotes, but pretty damn close. I mean, look at this. Mutation rates in cells, 10 to the negative 8, 10 to the negative 11th. That right there, 10 to the negative 8, that is a 1 in 10 billionth chance. Or no, sorry, 10 to the 8th, that's 100 million. 1 in 100 million chance. Or 1 in 100 billionth chance. So for every 100 million or every 100 billion, there may be one nucleotide mistake. Now, I'm not saying as it's going along, it's not making mistakes. It just turns out that in that replosome, there's going to be enzymes or parts of the enzymes that are doing a proofreading. It means they go back and they fix. So this 10 to the negative 8, 10 to the negative 11th chance is things that slipped through, that the proofreading did not catch. Turns out during the replication, Pol 1, Pol 3 are looking for what is referred to as mismatch. Instead of the A to the T to the G to the C, it's A to the G or A to the C, which is not going to pair up correctly and is going to result in a bulge, a distortion of the double helix, either the major groove or the minor groove, which is what these enzymes are just moving up and down, the major and the minor groove. And whenever there's a bulge, that means there's a mistake. They cut it out. Turns out that they know which is the template, which is not the template due to methylation patterns. Turns out before being copied, being replicated, both strands of the double helix will have methyl groups attached to certain nucleotides. So that way enzymes will quote unquote recognize which is the template, which is the new strand, and then fix, replace the mismatch on the new strand while leaving the older template strand where it needs to be. So Pol 3 built within it is a proofreading capabilities based on maintaining major minor groove, maintaining two parallel strands. If there's ever a mismatch, like here it's showing A to C, a bulge, a distortion in the shape, it halts, it cuts, it removes, it lays down the new, and then it moves on.